Hi, good morning, everybody. Welcome to the Advances in Research and Innovation stream of SNOMED CT Expo 2023. My name is Terence Sherd. I'm the Technical Operations Manager for SNOMED International, and I'll be your moderator for today. All questions will be answered at the conclusion of the presentation. Those of you online, if you want to ask any questions, please use the Q&A function at the bottom of your toolbar. Those in the room, there is a microphone behind the camera. If you could use that for answering your questions later. I'm now pleased to introduce Christine Spisla from Deloitte, who will be presenting modeling in vitro diagnostic device data in SNOMED CT. Christine, please go ahead.
Thank you very much. Hello, everyone. Like Terence said, my name is Christine Spisla, and I'm working on the SHIELD project that Dr. Campbell spoke about this morning in the keynote address. The project I am working on has been funded by the the Food and Drug Administration in the United States. I'm not speaking on behalf of the Food and Drug Administration, and I'm not speaking on behalf of the SHIELD. I'm just sharing some of the work that our project has done. And my co-presenter, Dr. Scott Campbell, is down the hall somewhere educating his audience on the cancer checklist. So he's been a part of this project as well. Oops, it doesn't want to advance. Yeah, I am the green one. Oh, there we go. So the issues with interoperability are very well known, and the downstream effects of them are known as well. And it not only the causes of interoperability, but also the issues that it has with data going down the road as well. And that has downstream consequences as well. So when we look at it from a lab perspective, and you think you've got the lab test name and you've got the value, everything should be fine. And as Dr. Campbell talked about in this morning's presentation, that you can have lab values coming from across different organizations, or you can have them being messaged back and forth within the same institution. And they're messaging one thing, but that receiving institution or that receiving entity just can't interpret it. And why is that? A lot of times that is due to the medical devices or the lab devices or the in vitro devices that are used as part of that test model. For instance, when you're doing a prothrombin time or an INR or any kind of blood clotting, that test result, no matter what in vitro device, that test result is going to be the same no matter where it is. But if you have blood chemistry, if you have a thyroid function test, depending on the manufacturer, depending on the reagent, depending on the IVD device that's used, your reference ranges coming from that lab value may be different. And if you're sending that to a different organization, institution, or receiving body, they're not going to be able to interpret it or even aggregate it across their institution. And this happens exponentially if we're talking about public health issues, such as happened with the COVID-19 pandemic. And, the COVID-19 pandemic really showed us that we were unable to measure disease instance because of the different testing modalities that they had across the healthcare sphere. You had the point of care testing, and then you had the PCR testing, and you were comparing apples and oranges. And that really impacted our ability to understand the disease prevalence in a certain area. So to address this, the SHIELD project has been working on that as well. And the SHIELD project is an acronym, and it stands for the Systemic Harmonization and Interoperability Enhancement for Lab Data. And so that's why I'm just going to call it the SHIELD project. Initially, there was the LOINC2 IVD test, and that's the first step that they used to try and get some interoperability across lab data. And what that was the manufacturers provided the LOINC codes for their customers, and that worked great as long as everyone was working with the same manufacturer. And so that the livid, it focused on describing that same lab test the same way, but it was only for that same vendor. And Shield wants to go the next step. It wants to describe that same lab test the same way and be vendor agnostic. And so, like the slide says, lab data is very simple to transmit, but it's when you're aggregating it, when you're interpreting it, that's when the issues start to happen. And different manufacturers, different EHR vendors, they all kind of add another step of complexity to this equation. So when we're trying to create real world data for public health, for even the patient at the bedside or entire populations when we have the next pandemic, we need to look at how can we resolve this and how can we improve the interoperability of lab data to improve patient safety and public health reporting, but also research and innovation, as well as clinical decision support moving forward. So this is where Shield comes in, and Shield started working on this prior to the pandemic. And the FDA sponsored a program which became Shield, and they had over 70 organizations who were stakeholders in this. And I just want to give you a little bit of background here on the Shield program so you understand the work that we did. And really it's to improve and build and implement a comprehensive solution so that we can improve the interoperability of our lab data across the enterprises. And it not only addresses the challenges with lab data collection, but also with analysis as well. And this was started before the COVID-19 pandemic, and when the pandemic came around, it just provided the perfect use case for why this is so important. So before we talk about data interoperability, we really need to talk about quality lab data. And as Dr. Campbell said in his talk in the keynote this morning, you can't send bad data across the systems because that's going to, just sending bad data just proliferates the problem exponentially. And so as part of Shield, they looked at not only the interoperability, but improving lab data. And they created LEADER, which is the Lab In Vitro Diagnostic Repository. So it creates that single source of authoritative truth of what should be those necessary and sufficient lab data elements that should be transmitted across organizations. And as part of this was the creation of the Integrated Knowledge Management System, which Dr. Campbell spoke about this morning. And when I talk about it a little bit later on, I'm going to refer to it as Comet and Tinker. And if you've been down by the exhibits and been at the National Library of Medicine booth, and you can see just an amazing demonstration of what it can do. And so we need to improve the quality of the lab data that we're sending, and it's not only the way we send it, but what we send as well. And this involves not only the laboratories, but it involves our vendors as well. So in looking at how can we improve the lab data quality, the first thing we did is we looked at what is going on out in the industry, who's doing what, and what can we build upon. We don't need to reinvent the wheel. So looking at the bottom half of the screen, in the lower left-hand corner, in Australia, they've done a lot of work using one of the ISO models for the Therapeutic Goods Administration, and they've identified a number of data elements that can be used, but they focus on public health. And in the lower left-hand corner there, the Global Medical Device Nomenclature is using that same ISO standard, and they're creating naming structures for those medical devices. And in the upper right-hand corner, there's yet another ISO standard that's being used for the diagnostic medical devices. But this really talks about the different functionality between them. And it talks about the different components of them, and it provides names for the different reagents, the reagent strips, and all the different tools that the labs use to process our lab specimens. And then in the upper left-hand corner, I looked at SNOMED and what has SNOMED done already, and what can we leverage from SNOMED to create these attributes that we can define these IVD devices as we send our the lab results from organization to organization. And I looked at the physical object hierarchy. Now, currently, this hierarchy has no defining attributes that we can leverage. And they even say in their editorial documentation that in vitro devices are out of scope for this. But that doesn't mean we can't try and we can't start to see what's there and leverage what can be done. The SNOMED the guidance for the physical object hierarchy also says that they define what a device is, and it dovetails very nicely into what the lab devices are, because these are the devices that have no contact with the patient. So these aren't the crutches, these aren't the bandages, these aren't the assisted devices. These are what's in the lab. So I started to take a look at these leader data elements and what can we reuse. And on the left side of the screen, the bullet points that we have here actually fall under the test kit identification and the instrument identification themselves. And so I'm looking to see what is necessary and what is sufficient to help define these in vitro devices as part of that message that gets sent to as part of the lab results to the receiving organization. And we look at the product markers. We've got the product identifier. We've got it's skipping the lot number. There's all sorts of identifiers that come with the test kits that are used, not only the machine itself. And then the test kits can change from vendor to vendor. So some of these attributes that I identified might be very applicable from patient to patient, or they might be applicable to the patient population if you're doing public health, and they may not cross between point of care at the patient and public health. So again, we need to look to see what is truly necessary and sufficient. And then I look to see what other kind of defining attributes we can pull from SNOMED itself. And there are a number of defining attributes that are used in other hierarchies that we can reuse in the physical object hierarchy to try and define our IVD devices. And LOINC has codes that we can use as well. And looking at leader, there's a whole laundry list of other codes that we can use. But this is discussions that need to be started, and people need to come together and just start to talk about what they need and what they need to send and what they need to receive as part of these IVD devices. So first of all, I'd like to thank my colleague, Scott Campbell, for the screenshot here on the left-hand side. This came from the University of Nebraska, their SNOMED extension. And we can see that what they've taken some of these what they've done is they've taken some of the LOINC attributes and modeled them in SNOMED. And if we look at the right side of the screen, we can see some additional leader attributes that we can start to add to this concept as well. But then we need to stop and take a look, and do we want to overload a lab concept in SNOMED or a LOINC concept, overload it with defining attributes to include that IVD device data and what is going to be the downstream effects of that. So again, we need to figure out what is necessary and what is sufficient. But as we're trying to do this, we need to start to play around and see what's going to work and what isn't going to work. So the device hierarchy, there's been some work by SNOMED already to remodel the device hierarchy. And looking at the reuse of some of these attributes, it dovetails very nicely in with the project goals of redefining this device the SNOMED device hierarchy. And we need to look at what defining attributes SNOMED has and how, if we can reuse it and how it can be reused. And not only look at some of the defining attributes, but some of these unapproved defining attributes, because there's some gold in there. And we need to really kind of play around and see, you know, thinking outside the box and how can we reuse what's already out there to try and use this. And then also looking at some of the other ontologies out there that have the basic foundational or formal ontology, and also the relation ontology has a number of defining attributes that we can pull into SNOMED and also pull into the Link extension that we can use to model our IVD devices. So this is really kind of thinking outside the box and extending those editorial guidelines to see what can we do in order to meet this need of modeling the IVD devices. So as I look at the physical object hierarchy, it's like as I said, there's no defining attributes right now. And the one thing that we need to also look at is, do we need to send everything in one concept together, or can we send something separately? Last week in the LOINC meeting, they were talking about sending one of their properties as a separate message. So as we are sending our lab values across to either public health agencies or another healthcare institution, maybe thinking about not packing the IVD device information within that test result itself, but sending it as an additional field along with that. You know, this is the kind of thinking that needs to be done in the kind of outside of the box configurations to see what's going to work, and as well as what's not going to work, because that's going to give you just as much information. So this is where our integrated knowledge management environment comes in handy. This is where, and Dr. Campbell talked about this morning, because it provides an area and provides somewhere where different terminologies have come together and harmonized together, where we can start to play around between the different terminologies and see what works and what doesn't. And as we do that, we need to keep the high reliability organization principles and safe systems principles in perspective. When we initially, especially in the United States, went from the paper record to the electronic health record, there was a rush. The guidance we got from our vendors was just whatever you have on paper, just put it into your electronic record. We can fix it later. Well, it's been 20 years and a lot of these issues have yet to be fixed. And so this is where this integrated knowledge management can help assist with refining and optimizing our electronic health records. And also leveraging our SNOMED CT, RX norm, LOINC, and other resources that we have, like different extensions that people have worked on. So when we're talking about our integrated knowledge model, we need to also talk about that the common model to support the terminology. And so the integrated knowledge model that Dr. Campbell was talking about this morning, it supports the import, the editing, and the authoring of additional concepts and additional terminology, because it's not just enough to be able to import your terminologies and to be able to view them as how they kind of nestle among each other and how they harmonize, but you're being able to edit and make some changes and to see what the downstream effects of those changes are and see how the terminology evolves as you make those changes. And then also being able to author and include new concepts and see what the downstream effects of those new concepts are as you've added them to this integrated terminology as well. So the integrated knowledge model that we affectionately call COMET, the goals of this is to create one authoritative source to harmonize how lab data is viewed and how the standards come together and then can be pushed out further to the healthcare institutions. And also it improves the goal is to improve the public health reporting. In addition to that, healthcare research and innovation. And that's the important part, is the innovation, because you cannot do things the way you've always done them, because you're going to get the same thing over and over again. Sometimes when thinking outside the box, that's when, these, I hate to keep using the word innovations, but these new ideas or these new products or these new terminologies come around that really move healthcare forward. And that's what we're trying to do, because it's not a matter of if we're going to have another global pandemic, it's a matter of when. And if now we have lessons learned from the COVID-19 pandemic, if we can take those and then start to evolve our terminologies that we use for interoperability, we can be better prepared to monitor that next pandemic and then also move toward controlling it from a healthcare perspective. And then also the goal of our IKM is to integrate all these multiple information and terminology standards so that we can see what the equivalents are, because there are equivalents in many of these terminologies. They were created for a purpose, and then they themselves evolved, which created these redundancies. And to be able to incorporate the new releases of each of these terminology, or as these terminologies release asynchronously, you can still seamlessly incorporate them as well. So as part of COMET, we have a terminology architecture, which is called Tinker, because each of the terminologies, so RX norm, SNOMED, LOINC, they have different properties, and the different properties sometimes align, sometimes they don't. But what Tinker does is it has harmonized all of these kind of agnostically, and it puts them together seamlessly. And if you've been to the NLM booth downstairs, you can see how LOINC and SNOMED have integrated and the parent-child relationship that's been created. And that was done using this Tinker architecture. And this is how we can leverage this IKM, this integrated knowledge model, in order to find these redundancies and to clean up the terminology and to improve that data quality as part of our goal to improve lab interoperability. So some of the challenges that Tinker is designed to overcome, the inability to recognize equivalence. I know that it's not only within the terminology itself. I am a nurse. I've worked with nursing terminologies, and I know that, in healthcare, we love to say the same things about a dozen different ways, because the clinicians like to say what they like to say it, and they want to document it the way they want to document it. And so the Tinker architecture allows us to recognize these equivalents, and I've seen it in practice, and I think it's cool. It also allows us to identify concepts. So when the pandemic happened, being able to add additional concepts in real time and push them out and publish them, and being able to see where they lie in the architecture. But then also moving up to our point number one, when the pandemic happened, the name of COVID-19 evolved over time. And as they were adding these terms to the terminology, that name was evolving as well. And so that was the potential of getting or introducing duplicates in their terminology. And the Tinker architecture, using the harmonization of all the attributes across different terminologies, allows us to find those equivalents as we're very quickly adding new concepts in, and identifying that flawed information, and to be able to correct that as we find something that maybe was entered either erroneously, or the knowledge around that particular concept had changed over time. And then to be able to do that, to evolve the terminology safely and reliably over time. That is something that has challenged everyone over time. And what we don't want to have happen is have the terminology be responsible for any kind of a patient safety event. So the benefits of Tinker, like I said, the changes can be viewed immediately. So as you're authoring, as you're editing, as you're authoring new content within Tinker, there is a reasoner. So you click on the reasoner and it goes through a classifications process, and you can see immediately the effect that the change that you've had has been what it has on the terminology. And it has a rollback feature so that if something didn't go as planned, you're able to roll back to the next version. And as part of that, it has an audit trail, a very clearly defined audit trail. You knew who did what and when they did it. And that's something that's missing in a lot of the terminology that we work on, not only the big standardized terminologies, but in your own institution as well. You know, who did that? I know when I worked on mine, there was no audit trail. And any data source can be added. So it's not only restricted to the big ones, SNOMED, LOINC, and RxNorm. You can add your local extension. You can add your local data dictionary as well into this and see where that all fits in there as well. And you're able to create extensions faster, which is in a pandemic situation is really key, to be able to document that information and to share that across other entities and for public health data aggregation. So the takeaways of this. So there's a need for to improve the interoperability of healthcare data, and terminology is just one piece of the puzzle, as is adding the in vitro device data to improve patient care at the bedside, public health reporting, and research as well. And as we do this, we cannot lose sight of the safety of our patients. It needs to be at the forefront, not an afterthought. We need to leverage the work that's already been done. We don't need to reinvent the wheel. We don't need to start over. There's a lot of work out there. We need to find out what it is and how we can incorporate it. And innovation, it requires new way of doing things. You can't do it the way you've always done it. And Comet and Tinker right now are an amazing environment and a way to address the silo development of terminologies that's happened throughout time, and how to manage these terminologies together in one environment, while respecting the unique properties of each of them as well. So with that being said, take any questions. Thank you.
Thank you, Christine. We've run out of times online to do any questions. I'm sure Christine would be happy to take any questions after the session from anyone in the room. We have no online questions, so that's okay. So please feel free to collar Christine after. Thank you very much, Christine. That was a great session. Online delegates can join another session through the event lobby, and that'll be starting in about 1 minute. Thank you.